In this video, we will talk about the membranes of the heart and the wall of the heart. We understand that we have in our body, we have the serosa membrane. And if you remember this serosa membrane, there was two layers. There was the parietal layer, which is away from the organ, and then there was the visceral layer. And between them, there was cavity, usually filled with the fluid. The idea of the serosa membrane was, if you remember, to reduce friction and therefore to protect the organ. So we have a serosa membrane for the heart. However, this serosa membrane, we call it the pericardium. The pericardium. And in general, we consider that the heart is sitting within a precardial sac, the precardial sac. So what are those layers of the pericardium? The outer layer, the very outer layer of the heart, we call it the fibrous pericardium. Attached to it and fused with it is the parietal layer of the serosa pericardium. So technically, those layers are fused together. The fibrous pericardium is dense connective tissue, and the parietal layer also connective tissue, and they are fused together, and both will form the outer layer of the heart. Then we have the cavity, the pericardial cavity. So this is usually filled with the fluid. Then we have the visceral layer of the serosa pericardium. Remember, we said we have a parietal layer and visceral layer. This was the parietal layer, and now this is the visceral layer. But this visceral layer also is fused with the heart wall itself. So therefore, we will consider this as the first layer of the heart wall. So the first layer of the heart wall is the epicardium, which is the visceral layer of serosa pericardium. Okay? Then the second layer, which is the thickest layer of the heart, is the myocardium. Myocardium, which is basically cardiac muscles. And the third layer of the heart wall is the endocardium. This is endothelial cells, a form of epithelial cells that line the inner cavities of the heart. So it lines the arterial and vein. The special thing about this layer that is continuous with the lining of the blood vessels. So why do we need a lining for the cavities of the heart? Because we need a smooth blood current in the heart. I don't want waves, because waves will create areas where proteins might settle, and I don't want that. So the smooth surface, it ensures that the flow or everything is running along with the blood, and it's continuous with the blood vessels, because I don't want a dent. Think, for example, this is the aorta and here is the ventricle. So here I don't want this dent or this area, therefore it will be like one straight line. Think of it like a carpet between two rooms. A carpet between two rooms, to make that transition smooth between the two areas. So that's the idea of it continuous with the blood vessels. Okay, so this is the heart wall. Other structures that we need to be aware of in the heart is, the first one is the area between the two ventricles. The area between the two ventricles, which is this. This is an area called interventricular septum. Interventricular septum. This separates the left and right ventricles. Okay, this is one thing we need to be aware of. The other thing, that inside the ventricles, we have a group of muscles. Those muscles are called papillary muscles. Papillary muscles. So those muscles are special in the ventricles. We will see them in the lab when we dissect the heart. Those muscles hold tendons. Those tendons are called chordae tendineae. The chordae tendineae. And those chordae tendineae attach or connect, maybe attach is better, attach the flaps of the valves. The cuspid valves. See how? This is the flap of the valve. So they're attaching it. Okay? The purpose is to help in preventing the backflow. Technically, those would prevent the flaps from opening toward the atria. So the flaps always will open toward the ventricle. Okay? I don't want them to open the other direction, because if they open the other direction, it means that the blood will go into that direction, toward the atria from the ventricle, which I don't want that. So those chordae tendineae and the papillary muscles will hold the flaps in place when the blood is pushed out of the ventricles. The third thing I want you to understand also is the thickness of the wall between the left and the right ventricles. The left ventricle is thicker than the right ventricle. Why it's thicker? Because it has to send the blood to the systemic circulation, which we said we need high blood pressure. So the extra thickness is to increase the power of the pump. So when the heart pumps, we have much stronger push from here comparing to the right ventricle. Right ventricle, if you remember, we said it sends blood to the lungs, to the pulmonary circulation, which we said we don't need high blood pressure. Actually, it's low blood pressure. Okay? So this is also a difference between the left and the right ventricles. And this is, we will see it in the lab when we dissect the heart. Another thing we want to talk about is back to the valves. We need to understand how do they close. So we already mentioned that the flaps of the cuspid valves are held by the chordae tendineae. So when the blood is flowing through the atrium, it's easy to open because the blood flow here, because they will open because the weight of the blood. So when I have enough blood here, and we will talk about it during the cardiac cycle, when I have enough blood here, they will open, and therefore the blood would rush through. Okay? But then when the heart start to contract, it means we are pushing the blood away from the ventricle. Technically, it has to go to the aorta. But in this case, I need to close those flaps. So what's going to happen is this. When the blood is pushed away, the semilunar valve will open. And here, those flaps will try to open to the other direction because of too much blood or too much power, too much blood pressure. So the chordae tendineae will try to hold them in place. So for the valves, the mitral valve and also for the tricuspid valve, both of them, the back flow of them or the opening into the other direction or into the opposite direction is prevented by the chordae tendineae. Okay? You would think of it like there is a door and you are holding, or someone is holding the door by their hand. So the door will not go in that direction. The door will only open in toward the person. Okay? So those are the mechanism, or this is the mechanism of how the mitral valve and the tricuspid valve are prevented from opening into the atria and therefore preventing the backflow. Now, let's talk about the semilunar valves. For the semilunar valves, when we are filling the ventricle, the semilunar valves will be closed. Technically, those are closed for two reasons. Okay? First one, not enough blood pressure from ventricles. Okay? Those, in order to open, we need so much blood pressure here. Think of it like you have two doors or a door. You see like old Western movies when the guy would come through those two doors, the flaps. So this is exactly, those are the semilunar valves. Okay, so if I have enough blood pressure, they will open. Okay, but now they are closed because there is no enough blood pressure. This is one reason. And to understand the second reason, when we have this blood pressure opening them, okay, the ventricle will contract for maybe half a second, then it will relax. So we will lose this blood pressure again. Now this blood, I need you to think of this blood that was already sent into the aorta. This blood will try to go back because of the gravity. But when it goes back, it falls into those sockets. The sockets that are formed because of the flaps. So it will fall into them and it will fill them. And once it fills them, it will seal the two flaps together, kind of sealing them, and therefore preventing the blood from backflow. So those flaps on their backside or on their aorta side, they will form pockets. Those pockets will be filled with the blood, and that will help in closing them because of the pressure on those flaps to seal with each other. So that's the mechanism of how those flaps are going to close. Okay, so for the aortic valves, you can write it. Aortic cuspid valves open because of blood weight. Okay, backflow in them is prevented because of chordae tendineae. This is for the cuspid valves. both the tricuspid and the bicuspid. For the aortic valves or the semilunar valves, open during ventricle contractions due to high blood pressure from ventricles. This is how they open, and close when blood in aorta and pulmonary trunk tries to go back to ventricles. It fills the bucket in the aorta and pulmonary trunk. Those buckets formed by the flaps of the valve. Okay, so those are the two mechanisms of how the valves are open and close. One thing also I need you to understand is that both cuspid valves are open and closed at the same time, and both semilunar valves open and close at the same time. Okay, so there is a high coordination between the left and right groups of valves. So the cuspid valves will open and close at the same time. Semilunar valves will open and close at the same time. But I will have either of the groups is open. So if the semilunar valves are open, it means the cuspid valves will be closed. If the cuspid valves are open, the semilunar valves will be closed. We cannot open the 4 valves all together at the same time. No, either I'm having the cuspid valves open or the semilunar valves open. So sometimes we will get some disorders in those valves. So they will lose their ability to close completely, okay, or open completely. So those will turn into incompetent valves, and that will cause valvular heart disease. And this is like a spectrum of diseases from benign to lethal. Some of them they can cause death. Okay? So the idea is that if those valves are not closed completely, it means there is a backflow of the blood. Or if they are not opening completely, it means the heart, it has to perform extra power or to do extra force to push the blood away from the ventricles. Okay? Over time, sometimes they might calcify and therefore lose their flexibility. So treatments for them sometimes will be like replacing them completely. Okay, either with synthetic valves or valves from animals. Okay, but those are like, a source of problems in the heart. So this is the pathway of the blood through the heart. Again, this is what we draw in the first video. And as I said, you need to master it. I will stop this video. In the following video, we'll talk about the coronary circulation.